White blood cells, or leukocytes, are mobile immune units, specialized delivery vehicles navigating the bloodstream to execute targeted chemical destruction. These circulating defenders are broadly divided into two distinct morphological categories based on what they carry inside their cytoplasm: the granulocytes and the agranulocytes. Granulocytes are defined by two physical traits: highly lobulated segmented nuclei and dense, easily stainable vesicles, or granules, packed within their cell fluid. This group includes neutrophils, eosinophils, and basophils. On the other hand, the agranulocytes, which include lymphocytes and monocytes, lack these easily visible specific granules and possess solid, non-lobulated nuclei. Understanding immune defense requires viewing these distinct shapes, the nuclear lobulation and granular payloads, as functional solutions to the problems of tissue penetration and pathogen neutralization. This diagram shows a comparative cross-section of two distinct white blood cells. On the left is the neutrophil. Measuring 10 to 12 micrometers in diameter, its defining feature is a heavily lobulated nucleus divided into 2 to 5 distinct segments. This segmentation provides extreme physical flexibility, allowing the entire cell to contort and squeeze through tight spaces. Surrounding that nucleus is an arsenal of chemical payloads. The cytoplasm is packed with secondary specific granules containing lactoferrin alongside primary azurophilic granules. These primary granules are essentially lysosomes loaded with the enzyme myeloperoxidase. Moving to the right side of the diagram, we have the monocyte. At 14 to 18 micrometers, it is the largest circulating leukocyte in the bloodstream. Unlike the neutrophil, the monocyte possesses a single large, eccentric, horseshoe-shaped nucleus. This is surrounded by an extensive, clear cytoplasm that presents a frosted glass appearance. This cytoplasmic volume houses the necessary organelles the monocyte will need to eventually exit the blood and differentiate into a massive, highly active tissue macrophage. Within the bloodstream, these cells are not active combatants. They are pre-engineered payloads, circulating passively until a chemical trigger signals their deployment into the tissues. When a localized bacterial infection takes hold in the tissue just beneath a postcapillary venule, that chemical trigger is fired. Through chemotaxis, white blood cells are actively guided toward the site of infection. They follow a concentration gradient of inflammatory mediators, molecules like interleukin-8, complement factor C5a, and leukotriene B4. This diagram illustrates the first mechanical phase of exiting the bloodstream, rolling. A circulating neutrophil makes contact with the endothelial wall of the venule. The leukocyte begins to roll along the endothelium by transiently binding to E-selectins. These receptor molecules, expressed on the surface of the endothelial cells, slow the leukocyte's momentum. Next comes the adhesion phase. The rolling stops when leukocyte integrins, specifically LFA1, make a tight connection with ICAM1 molecules on the endothelium, located at the junctions between endothelial cells, guide the leukocyte out of the blood vessel. The segmented nucleus acts as a series of hinges, allowing the entire cell body to compress and slide through the tight, intact endothelial junctions. When this receptor handshake fails, it leads to the pathology known as leukocyte adhesion deficiency, or LAD. LAD is caused by a genetic defect in the integrins. Because the neutrophils cannot execute the adhesion phase, they remain trapped in the blood. Patients suffer severe bacterial infections, devoid of pus, because the primary defenders cannot reach the tissue. Exiting the bloodstream is a restricted, receptor-gated barrier. This sequence ensures that inflammatory cells deploy only where they are actively needed, preventing damage to healthy tissue. Once in the tissue, the neutrophil crawls forward via amoeboid movement, protruding its cytoplasm to navigate the extracellular matrix. This diagram highlights the mechanical steps of phagocytosis. The neutrophil recognizes the bacterium, folds its outer membrane inward, and engulfs the threat into an isolated internal vesicle known as a phagosome. Immediately, the neutrophil's primary azurophilic granules mobilize. They fuse with the phagosome membrane, dumping their chemical payloads directly onto the trapped bacterium to create a phagolysosome. This triggers the respiratory burst, a chemical reaction unleashed exclusively within the confines of this isolated organelle. The biochemistry is precise. The enzyme myeloperoxidase utilizes hydrogen peroxide and chloride ions to synthesize hypochlorous acid, dismantling the pathogen. When a severe systemic bacterial infection occurs, the demand for active neutrophils outpaces the bone marrow's standard maturation rate. In these high-stress states, circulating neutrophils exhibit toxic granulation. As seen in this histological view, the primary granules become coarse and dark blue. Clinicians also look for a left shift. Under extreme marrow demand, the body prematurely releases unsegmented band neutrophils into the blood before their nuclei have fully matured. Isolating these chemical reactions within specialized organelles prevents the phagocyte from inadvertently digesting its own structural proteins. Following the initial assault by the neutrophils, the monocytes arrive. Once they exit the bloodstream, they undergo differentiation, expanding into massive, highly active tissue macrophages. While a neutrophil survives in the tissue for 2 to 5 days, a tissue macrophage can live in the interstitium for months. Acting as the heavy-duty scavengers of the body, macrophages take on the primary cleanup function. They phagocytose cellular debris and clear out senescent red blood cells. The macrophage also acts as a bridge to the adaptive immune system. This diagram highlights an empty major histocompatibility complex 2, or MHC2, receptor on the macrophage's surface membrane. After destroying a pathogen, the macrophage preserves specific fragments, or antigens, of the invader. It transports these fragments to the surface and binds them into the cleft of the MHC2 receptor. Displaying this antigen on the MHC2 receptor is the molecular signal required to activate targeted T lymphocytes. Macrophages resolve localized inflammation, but by processing and displaying pieces of the enemy, they also provide the biological intelligence required to trigger a highly specific, systemic adaptive response. Bacterial invasion is only one threat. To combat distinct pathological challenges, the body relies on different morphologies. Eosinophils utilize granules packed with major basic protein. Because large parasites cannot be phagocytosed, the eosinophil degranulates externally, bombarding the target directly. Basophils mediate immediate allergic hypersensitivity. They rapidly release storage vesicles of histamine and heparin to cause intense vasodilation and flood the tissue with immune factors. Lymphocytes, the agranulocytes, have massive nuclei for data processing and protein synthesis rather than phagocytosis. They produce the specific antibodies and cytokines that provide lifelong adaptive immunity. A leukocyte's specific morphology, its nuclear segmentation, overall volume, and granular contents, strictly dictates its physical capabilities. When clinicians run total white blood cell counts or differential blood smears, they are pulling direct diagnostic readouts of this mechanical and biochemical cascade reacting in real time. Each leukocyte's specific architecture, from nuclear hinges to chemical storage vesicles, is the mechanical requirement for its role in the body's defense.